And that's what we're digging into today. We have this transcript from Metabolism Made Easy that breaks it down.
The 3 stages of catabolism.
Yes. And the mission for this deep dive isn't just to list the steps. I think most of our listeners probably remember the basics from biology class. We want to understand the why behind the architecture. Why does the body move molecules across certain membranes? Why are some reactions isolated in the mitochondria while others aren't?
That spatial organization is everything. It's key. Biology isn't just about the chemistry, it's about the geography. Where a reaction happens is just as important as the reaction itself.
So let's trace the journey. We're going to follow a food molecule from the moment it enters your gut until it literally powers a thought.
Or a muscle contraction.
And the source material divides this into 3 distinct stages. So stage 1, it calls the great breakdown, happening in the intestinal lumen.
Right. And for this audience, we don't need to, belabor the point that proteins become amino acids or that polysaccharides become glucose.
Everyone knows that.
There is a nuance in stage 1 about fats, triacylglycerols that I think often gets glossed over.
This is the part that actually caught my attention. Because with glucose and amino acids, the logistics seem pretty straightforward.
They are.
You break them down, the enterocytes, the cells lining your intestine, grab them, and they just hop into the bloodstream. They're water soluble.
They fit right in with the plasma. It's a free pass. But lipids are hydrophobic. They hate water.
Right.
So you have this basic engineering problem. How do you transport a fat-based fuel through a water-based system like your blood?
And the solution the body came up with is, well, frankly, it seems inefficient at first. We break the triacylglycerols down into fatty acids and monoacylglycerols in the gut, but we don't just ship those pieces out.
No. Once they get inside the intestinal cell, we actually reassemble them. We spend energy to put the triacylglycerol back together.
See, that's the part that feels counterintuitive. Why smash a Lego castle just to rebuild the exact same castle inside the next room?
It's a packaging requirement. If you just dumped raw fatty acids into your blood, they'd clump together or even damage the capillaries.
Okay.
So the cell rebuilds the fat and then wraps it in a special protein lipid coat. It effectively builds a submarine.
The chylomicron.
The chylomicron. It's a massive, well, relatively speaking, lipoprotein particle. It encapsulates the hydrophobic fats so they can travel safely through the blood and lymph.
So that inefficiency of rebuilding the fat is really just the price of admission for using the bloodstream.
Exactly. It's a necessary cost for safe transport. And that really concludes stage 1. We've broken things down, and in the case of fats, packaged them up. Everything is now in circulation.
We are moving from the macro logistics of the blood into the microfactory of an individual cell.
And this is where the geography gets really, really interesting. So we've entered the cell, we're in the cytoplasm, but the ultimate goal here is to get to the mitochondria.
But not everything happens in the mitochondria right away. The source points out a split here. We've got 4 major pathways to talk about.
Glycolysis, pyruvate dehydrogenase, the TCA cycle, and beta-oxidation.
And only one of those is happening out in the lobby of the cell, the cytoplasm.
That's glycolysis. It's an incredibly ancient pathway. It's happening in the cytosol, and it's handling that glucose we absorbed earlier.
And this acts as the bridge, right? Because glucose is a 6-carbon molecule. It's too big to just waltz into the mitochondrial machinery as is.
Right. Glycolysis literally means splitting sugar. We snap that 6-carbon glucose into 2 3-carbon molecules. Pyruvate, and pyruvate is the ticket. That's the molecule that effectively knocks on the door of the mitochondria and says, okay, I'm ready for processing.
So glycolysis is the prep work. It's like chopping the vegetables before you put them in the oven.
That's a great analogy.
So let's talk about the oven, the mitochondria. The source material really emphasizes that the other 3 pathways, pyruvate dehydrogenase, TCA, and beta-oxidation, are all strictly mitochondrial.
They are deep inside in what's called the mitochondrial matrix, and there's a very good reason for that sequestration. These pathways are the real heavy lifters. They're stripping electrons off our food molecules.
Okay, let's break down what these 3 actually do, because the names can be a mouthful. Let's start with pyruvate dehydrogenase. We've got our pyruvate from glycolysis. It gets into the mitochondria. Then what?
Pyruvate dehydrogenase is kind of like the gatekeeper. It's a complex that takes that pyruvate, clips a carbon off it.
Which we breathe out as CO2.
We breathe it out as CO2, and it converts the remaining 2-carbon piece into acetyl-CoA. And acetyl-CoA is the universal fuel for the next step.
So it converts the raw delivery into the specific fuel type the engine can run on. What about the fats? the ones that arrived in those chylomicrons?
They go through beta-oxidation, and you can think of beta-oxidation like a salami slicer.
Salami slicer.
Yes. Fatty acids are these really long chains of carbon.
Yeah.
Beta-oxidation just cycles through and chops them into 2 carbon units over and over again.
Okay.
And guess what those 2 carbon units are?
Let me guess. Acetyl-CoA.
Exactly. So whether you ate bread, which is glucose, or cheese, which is fat, the cell funnels them both toward this one central molecule, acetyl-CoA.
Now, that is efficient. It standardizes the fuel source. So now we've got a big pile of acetyl-CoA sitting in the mitochondrial matrix. This feeds into the famous TCA cycle.
Or the Krebs cycle.
Right. What is the actual function of this cycle? I feel like people memorize the steps but completely miss the point.
The point is harvesting electrons. The TCA cycle tears that acetyl-CoA apart carbon by carbon. And as it breaks those bonds, it releases high-energy electrons.
But you can't just have electrons flying around loose. That sounds dangerous.
It is dangerous. It's basically radiation, so you need something to catch them.
Enter the carriers. The source highlights NADH and FADH2.
Right. These are the electron carriers. Think of them as buckets. The TCA cycle and beta-oxidation are just filling these buckets with high-energy electrons. NAD+ becomes NADH, FAD becomes FADH2.
And this explains the location, doesn't it? The source mentioned that having these pathways in the matrix is crucial. If this was all happening out in the cytoplasm, those electrons might react with the wrong things.
Absolutely. By keeping acetyl-CoA production, the TCA cycle, and beta-oxidation all locked in the matrix, the cell creates this really high concentration of these electron carriers, NADH and FADH2, exactly where they are needed for the next step.
It's just good inventory management. You're building up a massive stockpile of charged batteries right next to the device that's going to use them.
That's the perfect transition to stage 3.
Okay.
We have now finished stage 2. We have successfully turned our food into these high-energy electron carriers, NADH and FADH2, but we still don't have ATP.
We haven't paid the bills yet.
Not yet.
We have potential energy, but not usable currency. So stage 3 is the payoff.
That's the perfect transition to stage 3.
Okay.
We have now finished stage 2. We have successfully turned our food into these high-energy electron carriers, NADH and FADH2, but we still don't have ATP.
We haven't paid the bills yet.
Not yet.
We have potential energy, but not usable currency. So stage 3 is the payoff.
Oh, yeah.
And the location shifts again, just slightly. We move from the matrix to the inner mitochondrial membrane.
The barrier itself. This is where the magic, or, well, the biophysics happens. The source describes this as a coupled process.
A coupled process.
Involving electron transport and oxidative phosphorylation.
I've always found the word coupled interesting there. It implies some kind of mechanical linkage.
It is mechanical. It's one of the few places in biology where we see a literal rotary motor. But let's start with the electron transport chain.
Okay, so we have our NADH and FADH2 floating in the matrix. They approach this inner membrane.
And they hand off those high-energy electrons to a series of protein complexes embedded in the membrane. This is the chain.
Okay.
As the electrons get passed from one protein to the next, like a hot potato, they lose a little bit of energy.
And the cell doesn't waste that energy.
Not at all. The protein complexes use that energy from the electron to pump protons, hydrogen ions, out of the matrix and across the membrane.
So they are actively pushing protons against their concentration gradient, like pumping water up into a reservoir behind a dam.
That is the perfect analogy. You are building up massive electrochemical pressure behind that dam, the inner membrane.
And the electron.
The electrons eventually finish their journey by meeting up with oxygen.
Which is why we breathe. Huh?
It is literally the only reason we breathe oxygen, to act as the final trash can for those used up electrons at the very end of the chain.
So now we have this dam full of protons. We've essentially created a battery, a charge difference across the membrane. This brings us to the second half.
Oxidative phosphorylation.
Right.
This is where the turbine comes in. It's a protein channel called ATP synthase, and it lets those trapped protons flow back into the matrix.
They rush back through the gate.
And as they rush through, the physical force of their movement literally spins part of the ATP synthase molecule. And that spinning motion jams a phosphate group onto ADP to create ATP.
It effectively recharges the cell's main battery. That is the very definition of coupled.
It is.
The electron transport chain builds the dam, and oxidative phosphorylation is the turbine that generates the electricity from the water flood.
You cannot have one without the other. If you stop the electron flow, like with cyanide, the pumping stops, the dam empties, and ATP production just dies.
And if you poke a hole in the dam...
On uncoupler, yes. The electrons flow, but no pressure builds up, so no ATP is made.
Sure.
All that energy is just lost as heat.
It's a delicate, incredible system. And it brings us right back to the source's point about the high-energy molecules being readily accessible. Because stage 2 filled the matrix with NADH, the electron transport chain in the membrane has a constant fuel supply right at its fingertips.
The proximity ensures speed. When you are, say, running for a bus or even just thinking through a complex problem, your demand for ATP skyrockets. You don't want your NADH commuting from the other side of the cell.
You need it right there.
You need it right there at the membrane interface, ready to go.
So to zoom out and look at the whole picture, we start with a sandwich, stage 1, we dismantle it in the gut, we solve that tricky hydrophobic transport problem with chylomicrons.
We get the fuel to the cell.
Stage 2, we process that fuel. Glycolysis preps the glucose, beta-oxidation preps the fats, the TCA cycle strips the electrons, and we end up with a matrix full of NADH and FADH2.
The charged carriers, the full buckets.
In stage 3, we cash them in. We use those electrons to put protons, turning a microscopic turbine to mint-fresh ATP.
And that ATP is what allows your heart to beat while we're having this very conversation.
It really puts the "you are what you eat" saying into a different light. It's more like you are what you efficiently catabolize.
And you are where you catabolize. The compartmentalization is just as impressive as the chemistry.
Thanks for listening, and we'll see you on the next Deep Dive.